So basically, it's not necessary for me to compute to actually to use the silhouette plot on the clusters, right? I can use any grouping, right? That's already existing in the data, right? Such grouping for this zoo data set is a grouping to the type of the animal, right? And here I see in the silhouette plots there are some negative silhouettes. So negative silhouettes would mean, right, that that there are some animals, right, that actually do not belong to this cluster at all, right? I mean, they do not belong to the group of mammals, right? But according to the structure of the data in Euclidean distance, these two should be in a different cluster. And what do you think these two are in the mammals? It's dolphin and porpoise. Okay, so so dolphin and porpoise, according to Euclidean distance, right, should not be mammals, right? It should be in a fish cluster. And also, I'm seeing here, these invertebrates it's a mixed group anyway. So the slug and the worm and the scorpion says, okay, they should be rather with the insects and not with invertebrates. Yeah. So the idea is that so if the clustering assignment, so if I have a blue cluster here and a red cluster here, so if my clustering assignment, or maybe not a clustering assignment, but the label assignment would be so that like on this figure, then of course this blue point would have a negative silhouette. It would say, okay, this group, I mean, this point does really not belong to the blue group, right? But it should rather be in the red group. So negative silhouettes are fine, right? They just indicate that something was assigned a group that should be assigned some other group. Okay. All right. Then Lazar writes, I'm not sure if this makes any sense, but could clustering be done on account of correlation? Probably not, right? Correlations are simply finding out which of the features are related, right? Whereas clustering tries to find not relation, but what is close together, right? There is a particular problem with the data, right? So because, I mean, and we are actually jumping to this now. So the problem is that often you find the things, so if you have many features, just like, I don't know, here in this case, right? I have many features, right? And maybe some of the features are completely related to each other, right? So maybe what is the these features are all categorical. Actually, to compute correlations, I would need to actually not discretize, continuize them. Sorry. So that's a trick such that so that I will make all the features numerical. So these features in this data set are actually they're 0 and 1, but they're basically saying yes and no, right? So they're essentially categorical. I need to turn them into numbers, and then once they're a number, then I can measure correlations. And here I have, right? And so here it looks like this, right? Oh, I have something to show you, something else that's cool. So basically, whether you lay eggs or you give milk, right, is heavily correlated in a negative way, right? So if you give milk, you don't lay eggs. There's only probably, there are probably some exceptions, but, or if you have a hair, you also give milk as a species, okay? So because we male are not there yet. And then if you have feathers, right, you have 2 legs, right? So this will be the correlations, right? I'm not seeing any, this does not help me in any way to find the clusters, but it would help me to decide which are the features that maybe, so if this is so heavily correlated, right, this data would do just as well if I remove the X. So if I don't have any column with the X, well, fine, because milk tells me everything, right? And no hair either, right? So hair, I don't need, I mean, in a sense, where I'm pointing, I can reduce the dimensionality of the data if the correlations are very strong. Okay. The neat thing that I can show you also, I don't know, I'm starting to enjoy this. I don't know if you get bored with me, tell me, but what I can do, so I'm going to do the stuff. I'm going to actually transpose the data. I'm going to turn the data around so that now, so now the objects of my interest will be the features, right? So the hair, the feathers, the eggs, the milk, and so on, right? And the columns will be actually the different measurements, different animals, right? So I can do this by I can do this simply because maybe I would like to cluster the features. Okay, so if I cluster the features, then I have to measure the distances between the features, and then I can do the hierarchical clustering, and then it's the word. This is it. Enumeration would be instead the feature name, right? Yeah, I don't know if this helps, right? But here I find out that hair and milk are correlated, right? And the cat size and the teeth are also. And then fins, whether you have fins or you're aquatic, is also correlated, and so on, right? Whether you breathe and have a backbone and tail is also correlated in some way, right? So that's another. So I turn it around, right? And that's another thing how to use clustering. Turns out that legs is far apart from everything. Okay, I'm not going to push on this, so because I never seen. I mean, this is the first time I see on this data set this graph, but you can do that. And of course, you can decide that maybe, right? Maybe you have only 3 sets of features altogether, right? And it's already because these are correlated, and there's legs that's uncorrelated with everything else. So maybe you can just reduce the number of features to 3, and this is what you can actually observe in a 3-dimensional graph, right? Or if we say, okay, I'm not interested in legs, maybe I can just, I don't know, take eggs and milk, and that would be it, right? Although I don't know, not sure. Okay, there are. So I need to go ahead and actually finally present you. I've been turning around about the stuff that I would like to show you. Maybe I'm going to stay with the zoo animals here. So maybe just as an idea, right? So in life, there are things, right, that live in many dimensions. Like maybe they live in so many. It's hard to draw many, but we can perhaps draw 3, right? Let's say the 3-dimensional data set. Maybe, I don't know, the hair and the eggs and the fins here, right? Or I don't know. I'm pretending that this would be the, or maybe rather like. I don't know, maybe rather like weight and height, and what else? The blood pressure, systolic blood pressure, let's say. Okay, and maybe this data, right? It's not random in a sense that it fills out the entire space, but maybe this data is somehow related. Maybe there are some correlations, right? That tells us that this is not just a mess, right? But that there is a certain direction where the data spreads. Maybe it spreads in this direction, a little bit in the other direction here, right? Okay, such that I can take the 3-dimensional data and then maybe take a 2-dimensional data plane, right? And project each of these data to this plane. Okay, so what do I gain by doing this? I gain because I can visualize this plane, right? Because if I project everything to 2-dimensional plane, I can visualize my data. Okay, I also decided that I'm gonna visualize this data in the two-dimensional plane where the data is spread the most. So in the direction of the data, right? I didn't choose some other projection because maybe then the data squashes together in the sort of in a plot that would be too crowded and I wouldn't be able to say anything about it, right? And this is not so infrequent, right? So the data often, I mean, this of course is obvious for the 3 dimensions. So I'm turning 3D into 2D, right? But what if it's possible if I'm having 20 dimensions, like in this zoo data set, right? And turning this into 2D, right? So this I cannot draw because 20-dimensional data is like, I don't know, maybe Nikola Tesla could do something with that, right? I can't, right? But maybe there's within these 20 dimensions, maybe there's this two-dimensional plane where the most of the data is spread. And then I can also, I mean, if this would be okay, right? Then I can visualize this data, right? Okay. So let me stay just for a little while with this idea of 3D to 2D, right? And show you, I don't know. So on Safari, I'll show you a night sky. Okay. So basically we live in 3 dimensions, right? We think, right? I mean, there are other theories, but I won't go there. And this is perhaps, I don't know, a night sky, right? So the way somebody took a photo, right? I hate how Google places it, but maybe this one, right? So there's a picture of the night sky, right? What do we see here? So what is on this picture?
Stars, lights of a city.
Yeah. So here, okay, something, right? But can you tell me? Okay, I'll be interested in the sky this time. So there's obviously a highway and there's a city. But what about the stars? I mean, can you tell me something about the stars in the sky? But there is a particular shape that I see, right? Sort of like a line. So, and I don't know, if I look at the other photos of the night sky, right, I also see some, well, there's this one, right? I don't know why these images are now so bad, right? But there's also a line, right? Basically, what I'm showing you, right, and what every photographer does, right, it's good because these objects that are there, right, they live in the 3 dimensions, right? And on these pictures, I have 2 dimensions only, right? So I have a projection, basically. A projection taken from Earth, right? A projection of our galaxy, basically, because this is a Milky Way that, so this cluster here, right? It's basically a Milky Way, right? Okay, so now my experiment goes like, if I type a Milky Way, right? Or better, right? So maybe, let's say I'm in a, oh, where is that? Okay, here, right? So basically, if a teacher, right, in a school, let's say you go to the primary school and they talk about the Milky Way, right? And would the teacher say that, okay, so teachers usually use models, although they don't say that, right? Because they try to simplify the concepts, just as we scientists do, right? Would a teacher come into a class and say, well, this is a Milky Way? Would that be okay? Probably no, right? Just to remind you, right? Because this is a way we see Milky Way, right? I mean, maybe it's hard if we live in the city, but just go outside on a clear night, right? And you will see that there's a line, right? I mean, it's not really straight because we are somewhere in between the Milky Way. So it's a bit fuzzy, right? So maybe, I don't know, so I'm going to destroy the simplicity of my model, right? But I would say that this is now a Milky Way, right? Okay, maybe, I don't know, you're still bothered with the line, right? But this is a Milky Way, right? And we are probably not happy with that, right? Because this is not, I mean, although this is the way we see it, right? This particular visualization does not convey the structure of the Milky Way sufficiently enough, right? It says that, okay, it's sort of like, from our viewpoint, it's sort of flat, right? But there's another presentation of the Milky Way, another another projection which is more informative, right? So if I type Milky Way now in the browser, right, I get this. Okay, I also get a line, right, sometimes, or a line here or a line here, right? But usually when a teacher would go into a class, they would say, oh, this is a Milky Way, right? This one. Okay, so sun is here. This is why we see it in a bit fuzzy way, because we are in between, right? If we would be further away, it would really be a line. So Milky Way is sort of like, not like this, right? But Milky Way is sort of like this, right? I don't know. There's one spiral, there's another spiral. I don't know how many spirals. I'm not an astronomer, but this will be closer to a presentation of the Milky Way, right? The way scientists think the Milky Way looks like. So, I mean, this is again a projection, but nobody took this photo, right? Because the technology for that doesn't exist yet, right? It's going to take a while for us to make a photo like this, right? But that's a model, right? So a model of the Milky Way. And I'm saying that this model is more informative. So if I have two models, right? Say model A and model B, right? Both of the models are correct, right? But model A reveals more about the Milky Way. Also because, so one reason for that is that the spread, right? So if I look at just as a window, right, of my data points, the spread of my data points in this visualization, so they're more spread around the 2 dimensions. Here, they're not spread enough because everything is just a line, right? So basically, in visualization A, I better see the structure of the data, okay? This actually goes back to what I mean, there's a reason why I'm telling you this. And that is, so in 3 dimensions, right? If I would like to project the stars in the Milky Way such that I get the most information, it would be the particular projection actually that is on a plane, right? That actually defines where the stars are spread the most. And this technique is called principal component analysis. So the technique that would actually prefer the visualization like the visualization A, it's called principal component analysis. Principal component analysis. And principal component analysis actually takes the data in any number of dimensions and finds first component where the data is spread the most, okay? And then it tries to find the second component, which is perpendicular to that component, where the data spreads the most. And it does so actually until it exhausts all the M components, where M is the number of dimensions that the data lays on. We are most often interested only for the first 2 components, and the idea why is that, okay, because we'd like to visualize the data, and that's it, okay? But we would also like, right, that these first 2 components embrace as much as variation in the data as possible, right? I mean, ideally, right, the first 2 components would actually describe 100% of the data, but that would mean that I don't know, even in 20-dimensional data space, all the data would lie on the plane, which is not the case, because usually it doesn't lie on the plane because they're not only, I mean, this would happen, right? So this would be just equal, right? If within the 20-dimensional data, as an example, you would have 10 features that are completely correlated, where the correlation would be 100%, and you would have 10 other features where they will be completely correlated with the correlation of 100%, right? And then this would mean that basically you have only 2 dimensions, okay? But this is not the case, right? You can see from the correlation plot that this is usually not the case. So we are trying to find out basically the directions where the features are most correlated. So So after half an hour, I finally managed to actually include now correlations within this. So I'm not where the clustering is, right? Okay, but what I'm telling you, right, is that in dimensionality reduction, the correlations actually help me to reduce the number of dimensions, even to the number 2, right? So what I'm gonna use, I'm gonna do the following, right? So there's this PCA widget, right? So I'm taking the zoo data using the PCA widget, and this widget actually tells me about the variance, right? It tells me that the first component, so I have, it tells me that I have, I think, 20 different features in the zoo data set. And that the first component explains already almost 30% of everything, right? Here's my question. If the data would be entirely random, so there would be no correlation between the features, right? How much of variation would be explained by the first component if you have 20 features? 5%, right? Okay, so there would be no correlation, right? I cannot squeeze the data in any way, right? So 5% of the data would be explained by the first component, right? And here I have that the first component explains almost 30%, right? Which is 6 times as much, right? So this data actually, I mean, this graph actually shows that some of the features need to be correlated, right? And then if I take the second component, the second component explains the remaining 20% of the data, right? So together they explain about half of the variance. So with only 2 dimensions, I basically can represent half of the variance of this data, right? I would need the other half to be exact, right? But maybe I can draw now the figure to see how the data is spread in this two-dimensional projection where I explain 50% of my data, right? So I'm going to do, of course, the two-dimensional projection will be the scatter plot, and I'm going to use in the scatter plot I'm going to use. So I need to select. Oh, that's already it, right? So so the axis is principal component 1 and principal component 2, and this is the figure. Okay. And I like this figure a lot because it tells me about the relations between different types of the animals, right? There are mammals, right? And here are fish. Okay, and I see that some of the mammals, eventually this here, right? These mammals are kind of closer to the fish than the other mammals, right? So let me see how close they are. I mean, what are these? Oh, dolphin, porpoise, seal, right? So basically the fishy mammals, right? Maybe I can do this. Okay. And there are some hardcore mammals here, right? They're like, okay, a girl, a hare, a vole. Oh, okay, there's some strange ones. Bear, opossum, right? There's something that goes towards the reptiles, right? And that is a platypus for some reason, right? There are fish here, right? So there's some cover. I mean, here it looks like there are only 3 points, but basically this fish had exactly the same values of the features, and this is why they were placed here, right? But there's something here that's close to the fish, right? Oh, it's a sea snake, all right? So, and here I mean, even if I would say, okay, I don't know about the type, right? If everything has the same color, right? I could actually say, oh, maybe there's a cluster here, right? And maybe there's a cluster here, and maybe there's a cluster here, and then maybe it's a cluster here and a cluster here. So I would say manually, right? There are perhaps 5 clusters in this data set, okay? And that's nice, right? Because we people, right? So I told you, right, that if the data would be only in 2 dimensions, right, we would not need machine learning or any kind of statistics, right? So I don't know. I can see that some people would object to that, but if we would, I mean, but I would say two-dimensional data sets, we stop doing machine learning or AI, and life becomes much simpler. But this is not the case, right? And bear in mind that this is just approximation, right? Because this the graph that I'm plotting here, right, actually contains only 50% of the entire data, right? The other 50% is hidden somewhere else, right? Okay. So people thought, all right, I mean, this is, so I can still plot the type. This is okay, right? But what about the other 50%, right? And they thought about another set of techniques. And I would like to actually show you two. So this, the principal component analysis, this is called the projection. So this is all important term. So it's called a projection. Because I'm really, right, I'm really taking the many dimensions and I'm projecting everything to the plane. Okay. So it's in terms of the math, it's even simpler. It's not a projection, but it's called a linear projection. Okay. And it's also very simple to compute. And mathematically, this is well defined and it's great and so on, right? But it's a projection. But then people thought about the other things. So the other thing is like sort of, it's something that Ida showed you yesterday, just a quick introduction, right? So they said, oh, well, so what if, so data maybe lives in the 20-dimensional world, or more, right? So basically what we have, right, is, so here is our data set, right? So the data objects are here, and we can take two data objects here. I mean, this is of course the table with the features, right? But we can take two data objects, say object A and B, and we can measure the distance. That's exactly what we did in hierarchical clustering, right? We measure the distance between objects, right? And let's call this distance uppercase D, ij. Ij refers to two objects, object I and object J, right? So they will be two animals, right? I measured the Euclidean distance between two animals, but in this 20-dimensional space, right? And then I want to construct a two-dimensional visualization. So 2D visualization, where I would place these points A and B, so sorry, I and J, such that this distance here would also be the distance in the two dimensions. So I will indicate this distance in the two dimensions as Dij, right? So basically, I would like, right, I can do this with 2 points. I would like Dij, so the distance measured in 20-dimensional space, to be equal to that in a two-dimensional space, okay? I can do that if I have 2 points, right? But I don't have only 2 points. I have, I don't know, I have n points here. So I goes from maybe n is equal to 100, let's say. So I goes to 1 to 100, and j goes from 1 to 100, okay? And I would like to not just put these points A and B such that their distance is kept the same, right? But all the other points, okay? So maybe there are all the other points that I would need to place them somehow such that for every pair of points, right? So maybe I'm taking this pair, right? For every pair of points, the dkl would be equal to the distance kl in the 20 dimensions, right? So can I preserve these distances such that all the distances in 2 dimensions match the distances in 20 dimensions? Okay, is this possible? And I would like to construct my 2-dimensional visualization such that the distances are kept the same as in 3 dimensions. Is this possible? So the answer is no, right? It would be possible for 3 points, right? Because in 3 points, I can always put a plane, right? And the distances are the same, right? But as long as there's the 4th point that is outside this plane, right, I can no longer keep all the distances. So what I really want here, right, is, okay, I say, well, this is not possible, right? But maybe what is possible, maybe I can have an approximation, right? Maybe the distance between them should not be large. And I don't care whether the difference between the distances in the 20 dimensions and the distances in 2 dimensions, I don't care if they really match, but actually I should go through all the possible pairs, and I would like to minimize the sum of all the differences between the distances. So basically I put the points here on the 2 dimensions such that I minimize this equation, okay? And this is not simple, right? So the algorithm works such that, so it puts the points somehow, right? And then sees, oh, maybe this point should move somewhere here. And if I move it somewhere here, then I get further minimization of this equation. So it's sort of iterative algorithm where it tries to move the points, and then it tries to converge in the place where this is minimal, right? So where the differences between the distances in the original space and in 2 dimensions is minimal. And of course, I have to tell you that, so I can actually rotate this visualization. I can just take it and rotate it, right? And it would still be the same. The distances will be still the same, right? So this minimization is invariant to rotation, right? I can actually rotate it in any way, right? Around, and then it will be still the same visualization, right? So this particular visualization doesn't care. I mean, I should not say that this is X and Y, right? I should not talk about the coordinates because the coordinates really don't matter, because I can still rotate it and it's still the same, right? It's not like PCA analysis where the first principal component is well-defined and the second one is well-defined. Okay, this particular visualization is called MDS, multi-dimensional scaling, or MDS. Okay, so let's see the MDS from the zoo, right? And then, okay, the MDS displays itself, right? And that's the MDS. And it's not too far away from the PCA, basically. So in the low dimensions, the PCA and the MDS will be very similar to each other, usually, right? So again, we have, again, I think this is a sea snake. Yeah. And again, we have, this is probably the porpoise and dolphin. And so it's kind of similar, right? It's also similar because it starts with the PCA and then, I don't know, maybe we can see some. So it starts somewhere, right? And then tries to position the points such that that equation that I showed you, the value of that is minimized, right? But usually, because you would like to run it such that this becomes sort of like deterministic, you usually start with the PCA. So this is the PCA, and then you start applying the MDS, and then you see that it slightly changes. Okay. So it's not too far away from the PCA. There's a problem with MDS. It's a huge problem, actually. And so is the problem with the PCA as well. Actually, this lecture for me would be way easier if I would have you in a class. So if I would have you in a class, right, I mean, students usually sit, I mean, there's a particular structure when you see the students, right, especially when they're like 20 or 30 or 40, right? Every time they come to the class, right? I mean, we see that as teachers, right? There's a particular structure. There's a particular formation of students. And we observe, right, that often there would be pairs or triplets or quadruplets of students that would sit together. It's always like that, right? So they would not always sit on the same bench, right? But they would be clustered together, right? And there would be another group that would be clustered together, right? And then some students like to sit, I don't know, away from each other, right? Just, I don't know, take the the maximum possible distance to the professor, right? And you see this kind of structure, right? We don't observe the distances, actually. We observe the neighborhoods. So when I go and teach, right, I would register who sits with whom, especially if I give like, I don't know, 10, 15 lectures, right? I would know exactly the formation of the class and maybe the social links, right? Because there would always be these patterns. I'm telling you this simply because we human are like things that are together. I mean, the patterns that we observe are not that something is far away. So I'm not registering that student, I don't know, Masha sits 5 meters away from I don't know, John, let's say, right? So this is not something that I would remember. I would remember that Masha sits with Micha in the same bench. I would remember that, right? Also, our eyes work like this, right? Our eyes have a very tunnel vision, right? And we see the things that are close together, right? But we usually don't relate the things that are, I don't know, on the opposite sides of everything, right? So here was the idea, right? The idea was that, so in the MDS, right? So if this was my, I'm just drawing the data like it would be in the 3 dimensions, right? So maybe the data looks like this, right? So the MDS keeps all distances, the multidimensional scaling, right? Even the distances like from here to here, right? It would try to put these 2 points, right, which are far away in the original space. It would try to put it in the visualization. It would try to put it far away from each other, right? Whereas we human, right, we don't care about that. We only care about proximity. So we care that if things that are close together here, they should be also close together in our visualization, right? Or these things, they should be close together here, right? And these 4, they should be close together here. We don't care actually whether the distances between these 3 groups are kept exactly as they were, right? We care about preserving the neighborhoods. So preserve the neighbors. That's exactly what we want also with clustering, okay? Because with clustering, we would like to observe the clusters, right? That the things that are close together. We don't care whether they're far away, but we care if they're close together. And there's a particular algorithm that does that, and it's called t-SNE. It's so S&E stands for stochastic. Stochastic means it's going to be initialized randomly, right? There's going to be some randomness in the way it computes the things. This N stands for neighbor, stochastic neighbor, and E stands for embedding. Okay. I think it stands for neighborhood, actually. Yeah. So basically, so this algorithm actually tries to preserve the neighbors, right? And the technique is not a projection, but it's called embedding, just like MDS, right? So this transformation from 20 dimensionality from 20 dimensions to 2 dimensions in MDS, it's not a projection. There's no recipe of how this was constructed, right? Other than there's this criteria function that is optimized. And this is why the MDS is not projection, but it's also embedding. So it tries to embed the points within the 2 dimensions such that this equation is minimized. And the same is t-SNE, right? t-SNE has just other philosophy, right? It tries to embed the points within the 2 dimensions such that the neighborhoods are kept together. All right, I need to show you how the t-SNE works on this data set. And it's not going to be, so there's not going to be a wow factor. This is how the t-SNE works here, actually. I would claim the t-SNE for this data looks a bit better because you can see the colors are not, there's no overlap here, right? There's, the groups are really well defined, right? There's a red group. Okay, so here's like the fish are here, right? These are the exceptional mammals, right? So it doesn't look too much different from dolphin, porpoise, sea lion, right? No wonder, right? So I think compared to the PCA, right? I don't know. I can't decide which. I can't decide which one is better. But with small data set, t-SNE doesn't shine. So with small data sets, I would use PCA. There's not big difference here. Okay, let me give another example. Let me go to a particular data set I have in a data sets widget. And I'm sorry, this data set comes from biology. So I have it under AML. So this data set contains 1,000 of, this is called bone marrow mononuclear cells. Basically, they draw blood from, they draw blood from one patient and one healthy human. And they checked what the genes do. So they took, from this blood, they took each and every cell. And for this, each and every cell, they checked how active are the genes in the cells, right? And the question here was that, can you actually classify the type of the cell from these measurements? I mean, how many different types of the cells are in, actually not in blood, but in bone marrow? And what I'm gonna do is the PCA, and then I'm gonna do the scatter plot here. So this is how the PCA works, right, on this data. How many clusters do you see? Yeah, so they may be 3 clusters, right? And they're not well defined, right? And the MDS will give me actually a very similar look to this one. Usually on large data set, PCAs are sort of like this triangular shape. I mean, they always spread somewhere and then they spread in the other direction. They often look like this, right? Look at the t-SNE now. So t-SNE takes a while. It's a slower method. Okay, I'm going to remove color. This is a completely different picture, right? And here I see that actually there's a cluster here, there's a cluster here, there's one cluster here, one cluster here, one cluster here, and there's a one cluster here, right? There are at least 1, 2, 3, 4, 5, 6 clusters in this data. And it's interesting, they really correspond to different cell types. And I no way I can find them here, right? And so I can even go to what I want to do is this one. So look at this cluster here, right? So there is this cluster here that basically is this one here in the PCA. And this cluster here, it's somewhere here, right? There's a heavy overlap in the PCA. And there's this cluster here. So basically, the PCA doesn't give me, I mean, the idea is if I want to do the clustering, if I want to observe the clusters in the data that has sufficient number of data instances, I should use t-SNE. Because t-SNE preserves the neighbors, okay? And it's actually the visualization technique to be used to find the clustering structure. So PCA preserves the variance, MDS preserves the distances, but the t-SNE preserves the neighborhoods, right? So these are what I told you are 3 today prevalent techniques for data visualization, for 2-dimensional data visualization. There's a 4th one that is very similar to t-SNE. It's called the UMAP. The that you will also find today in the paper. But anyone today that publishes in these big journals like Nature and Science that has large data sets, they would either use UMAP or t-SNE for visualization. And they would often compare it to PCA, and they would often find that t-SNE, of course, reveals the clustering structure.